Welcome to BioMed Research Digest 2026-0602, today's 12 stories. Story 1, Cancer Vaccine Sustains 49% Melanoma Reduction After 5 Years. Story 2, Daraxonrasib Emerges as the Star of ASCO 2026 RASolute 302 Phase 3 Trial. Story 3, Smart Drug That Strips Cancer Cells of Invisibility Cloak Shrinks Tumors in Trial. Story 4, A Personalized Cancer Vaccine That Fights Melanoma mRNA plus Keytruda Combination. Story 5, High-Precision Base Editing Therapy Demonstrates Durable VOC-Free Results in Sickle Cell Disease. Story 6, Cancer Immunotherapy 2025-2026, 13 new FDA approvals and first-ever treatments. Story 7, Neoadjuvant Immunotherapy in Locally Advanced Head and Neck Cancer, Keynote 689 Phase 3 Trial. Story 8, Unveiling the Shared Gray Matter Signature Between Alzheimer's and Parkinson's Disease. Story 9, FDA Lowers Drug Approval Standards Now Accepting Single Clinical Trials. Story 10, Site4 Drug Predicting Drug-Binding Target Sites with an AI Agent. Story 11, APLSuite, an integrated suite for CD4 plus T-cell epitope prediction via antigen processing likelihood. Story 12, Mechanics of Pandemics, 10 Fundamental Principles of Disease Transmission. A new AI tool called Site4 Drug is aiming to solve one of the most stubborn bottlenecks in early-stage drug discovery, figuring out exactly where on a protein a drug should bind. Developed by Taehin Kim and colleagues and submitted to arXiv in June 2026, the system was accepted at the ICML 2026 workshop on generative and agentic AI for biology, drawing attention from both the machine learning and computational biology communities. Here is why this matters. Most computational drug design tools focus on the question of what molecule will bind to a protein. But before you can answer that, you need to know where on the protein to even look. For many proteins, especially membrane proteins, this is far from obvious. Accessibility, topology, and chemical modifications on the protein surface all affect which regions are actually usable as drug targets. Site4 Drug is designed to answer that prior question. The system takes in multiple sources of biological evidence, including protein topology, hydropathy profiles, post-translational modifications, disulfide bonds, and sequence context, and outputs a ranked list of candidate binding sites along with evidence summaries, risk flags, and a traceable decision log so researchers can understand why each site was ranked the way it was. One of the most interesting features is that the agent does not require users to specify upfront what kind of drug they are developing. Whether the program ultimately leads to a small molecule, an antibody, or a peptide therapeutic, Site4 Drug recommends the most appropriate binding modality based on the same evidence it uses for site discovery. This reflects how experienced drug hunters actually think and could help teams make smarter decisions earlier in a program. The system is also specifically engineered to avoid a common pitfall, selecting binding pockets that look attractive on paper but are biologically blocked in practice due to modifications or structural inaccessibility. That said, the paper is still a preprint and was presented at a workshop rather than a major peer-reviewed venue. There are no published benchmarks comparing Site4 Drug against established pocket-finding tools, so independent validation will be critical before the research community can fully assess its real-world utility. Still, the conceptual framing is sound, and if the performance holds up under scrutiny, Site4 Drug could become a valuable early-stage companion tool in both academic and pharmaceutical drug discovery pipelines. A research team has released APLSuite, a new open-source software platform for predicting which fragments of a pathogen protein will trigger a CD4-positive T-cell immune response. Published on arXiv in June 2026 by Jairui Li and colleagues at Chilean University, the tool is aimed at both computational scientists and experimental immunologists working on vaccine design and immunotherapy development. To understand why this matters, you need to know a bit about how the immune system works. When your body encounters a pathogen, it does not just throw antibodies at the whole protein. Specialized immune cells called CD4-positive T cells recognize very specific short peptide fragments, called epitopes, that have been snipped out of the pathogen protein and displayed on the surface of other immune cells. The process of snipping and loading those fragments is called antigen processing, and it has a huge effect on which epitopes actually get seen by T cells. The problem is that most existing computational tools for predicting T-cell epitopes focus only on how well a peptide binds to the MHC class II molecule that presents it, and they largely ignore whether that peptide would even survive antigen processing in the first place. APLSuite addresses this gap through an algorithm called Antigen Processing Likelihood, or APL. It combines 4 distinct signals: how flexible different parts of the protein are, how accessible the surface is, how easily the protein unfolds, and how conserved the sequence is across related organisms. In terms of practical use, the suite is designed to be accessible at multiple levels: Computational researchers can interact with it through a Python client and REST APIs, while experimental immunologists without coding experience can use a graphical interface. The pipeline runs with GPU acceleration and reportedly completes predictions in minutes, which the authors say outpaces existing tools. The software is open source and deployable on both personal computers and cloud systems, which broadens its potential reach significantly. For pandemic preparedness efforts in particular, the ability to rapidly screen entire pathogen proteomes for immunogenic epitopes could be genuinely valuable. The main caveat right now is that this is an application note describing the software, not a benchmarking study. The community will need to see rigorous head-to-head comparisons against established tools before APLSuite can be confidently adopted for high-stakes applications like clinical vaccine development. A team of 27 researchers from across the natural and social sciences has published a synthesis paper on arXiv titled Mechanics of Pandemics, laying out what they argue are 10 fundamental principles that govern how disease outbreaks spread and are controlled. Submitted in late May 2026, the work draws lessons from COVID-19 and earlier pandemics to identify mechanisms they believe hold true regardless of the specific pathogen involved. The breadth of the team is notable. The authors include epidemiologists, mathematicians, physicists, physicians, political scientists, psychologists, and public health researchers. That disciplinary diversity is intentional. One of the core arguments of the paper is that pandemic dynamics cannot be understood through biology alone. Social trust, political context, behavioral responses, and communication all shape how outbreaks unfold and how well interventions work. The 10 mechanisms are described as grounded in natural laws or in the structural properties of human societies. The authors argue these principles are underappreciated despite having a substantial effect on outbreak trajectories. While the paper does not present new empirical data, it synthesizes existing knowledge across disciplines into what the authors hope will become a shared reference framework for preparedness planning. Why does this matter now? Despite the enormous scientific investment during COVID-19, many response failures were not due to lack of data, but due to misaligned expectations, poor communication, and institutional blind spots. A clearly articulated set of universal mechanisms could help policymakers, hospital administrators, and public health officials apply lessons from one pandemic to the next more systematically, rather than treating each outbreak as entirely novel. That said, important caveats apply. This is a preprint without formal peer review, and arXiv itself has posted the standard advisory urging caution about clinical guidance use. The claim that mechanisms are truly universal across diverse pathogens and societies is an ambitious one that will draw debate from epidemiologists who emphasize contextual heterogeneity. The paper's real value may ultimately lie less in any single mechanism and more in establishing a common language across disciplines, one that could make future pandemic science more coherent and faster to translate into policy. NPR reports on the 5-year follow-up data showing that the mRNA-based personalized cancer vaccine, combined with pembrolizumab, is highly effective at preventing melanoma recurrence. The study reinforces mRNA technology's promise beyond infectious disease vaccines. This approach tailors the vaccine to the specific mutational profile of each patient's tumor. CS206, a base-editing gene therapy for sickle cell disease (SCD), demonstrates durable suppression of red blood cell sickling and significant reduction in vaso-occlusive crises and hemolysis. Unlike CRISPR-based approaches, CS206 corrects a single base without introducing DNA double-strand breaks, reducing risks associated with off-target editing. The therapy represents an advance in precision over existing gene-editing SCD treatments. The Cancer Research Institute's 2026 Insights, an impact report documents 13 new FDA immunotherapy approvals in 2025 alone, including first-ever treatments for previously overlooked cancer types. The report also highlights accelerating globalization of oncology clinical trials and faster regulatory pathways. This provides a comprehensive landscape of where cancer immunotherapy stands entering 2026. The landmark Keynote 689 Phase 3 trial demonstrated that perioperative pembrolizumab significantly improved 36-month event-free survival in locally advanced head and neck cancer, 57.6% versus 46.4%. This is a peer-reviewed publication adding to the growing evidence base for neoadjuvant immunotherapy strategies. The results support a shift in the standard of care for this difficult-to-treat cancer. This preprint presents the first quantitative vertex-level analysis of shared gray matter structural changes between Alzheimer's disease (AD) and Parkinson's disease (PD), leveraging large MRI datasets and a novel statistical framework adapted from genetic correlation analysis. The study identifies overlapping neuroanatomical signatures that may underlie shared disease mechanisms. Findings could inform biomarker development and cross-disease therapeutic strategies. The FDA has announced a significant policy shift, now allowing many new drug approvals based on a single clinical trial rather than the previously standard requirement of two. This change accelerates potential access to new therapies, but raises concerns among scientists about evidentiary standards and long-term safety. The shift reflects broader changes to the FDA's drug approval framework in 2026. Site4 Drug is a new AI agent designed to predict the most druggable binding sites on proteins, including difficult membrane proteins where topology and post-translational modifications constrain targetable regions. The system outputs ranked lists of sites with explicit biological constraints and evidence summaries, addressing a key bottleneck in drug discovery. This represents an applied advance in AI-assisted drug design beyond standard pocket prediction. APLSuite is a computational tool that predicts CD4 plus T-cell epitopes by incorporating antigen processing likelihood, a factor often overlooked by existing machine learning models. Better epitope prediction is critical for vaccine design, cancer immunotherapy, and autoimmune disease research. The tool integrates antigen processing steps to improve binding specificity predictions. A multi-author interdisciplinary preprint synthesizes 10 fundamental mechanisms governing pandemic spread, drawing on COVID-19 and historical pandemics. The work aims to identify universal biological and social laws of disease transmission to improve preparedness. Authors include leading epidemiologists and computational disease scientists from Europe and Latin America.